Okay, class, so we just, you should have done your prime time this morning, and then you watched our introduction video about gene regulation in prokaryotic cells. So now we're going to do our main activity for the day. So again, you guys know I'm using Notability. I love it. I hope it works out for us today, too. Hope there's no kinks. All right, so you should have this on your desk. You have a printout version of this, and you should be able to annotate this as we go with me, okay? So it says prokaryotic gene regulation. Prokaryotes and eukaryotes both contain chromosomes. However, their chromosomes are organized differently, and that was one of your primetime questions. Prokaryotes have a single large blank chromosome, while eukaryotes have multiple blank chromosomes wrapped around each histone protein, okay? So the first blank is talking about prokaryotes and that what kind of single large chromosome do they have? They have, so let's just look right here. So this one is a eukaryotic piece of chromosome, and this is a prokaryote, okay, it's chromosome. So it's a circular, you want to write circular chromosome, while eukaryotes have multiple linear chromosomes wrapped around those histone proteins that we've learned about. Okay, so let's read on. It says eukaryotes have or use transcription factors to regulate gene expression. Okay, so we've already kind of covered that, but I'm going to highlight it just in case. Make sure you know that. Highlight it on your paper. And it says prokaryotes use operons. And that's what we're going to learn about or review today to regulate their gene expression. So let's learn about some operons. You should have already read your chapter. You should have some background knowledge of what an operon is. So it says an operon consists of a promoter, a blank, and the genes they control. So in the blank, we have to write the last thing that's missing that I haven't highlighted, which is an operator. Okay, they have 3 parts. If you'll take note, there's also another part out here to the left of the operon. Okay, we'll learn about that in just a sec. So it says upstream, which just means before it, and not part of the operon, is a regulatory gene, which is very important. The regulatory gene codes for a blank protein. The repressor protein is involved in regulating the operon. So this regulator gene is read by, is read in transcription, in mRNA, we know that. That was the previous chapter, and it's converted with translation into a repressor protein. Okay, so it codes for a repressor protein. All right, let's keep moving. It says the RNA polymerase attaches to the blank to begin transcription of the genes within the operon. So this is all color-coded. So if we look back up here, if we need to, we can look at this diagram of the operon and see that seafoam green color is the promoter, okay? The RNA polymerase binds to the promoter or attaches to the promoter. This is how transcription happens of these structural genes at the end. Okay, all those purple colors or blue colors at the end. When the operon is in the on state, remember we talked about this the other day. If it's on, the RNA polymerase transcribes a blank strand of mRNA that codes for blank different proteins. Okay, so what kind of RNA? It says it right here. A single strand, 1. Remember, RNA is single-stranded, and unlike your DNA, which is double-stranded, okay? It's a single strand of mRNA that codes for multiple different proteins. For example, those proteins may work together in a metabolic pathway or they might not. You know, they might go to different parts of the organism and do its job there. So if we look up here, these are multiple genes. This would be like one gene, this would be the second gene, and this will be the third gene, okay, in our example. And we've already went over how this mRNA is made and how these proteins are made, right? They're made in the organelle called the ribosome. That was in chapter 4. So all of these things, you have to learn all of these things in order to use that information. Okay, so after transcription, the ribosomes carry out blank to produce multiple proteins. This is easy. This is from another chapter. Remember, this is transcription. And this one is translation, making proteins. Okay, so let's write that. Translation. That's from an old chapter. So how do we regulate this operon? Okay, so we've figured out that the prokaryotic cells have an operon. Instead of the 5 gene regulations of a eukaryotic cell, they don't have that because they don't have a nucleus. So when we now know that they have these things called operons and we know the parts, but how do we regulate it? Because we just don't want them running, making these genes or reading these genes and making these proteins all the time. So we have to regulate it somehow, or the organism has to regulate it somehow. Okay. So the blank protein regulates the expression of the operon by attaching to the operator, which is in orange. Okay, so let's look at the diagram and see if we can figure this out. Nobody yell it out, just in your mind, I want you to think of it. What protein is repressing the operon? Pretty self-explanatory, right? The repressor protein. I'm going to zoom back out and I'm going to ask you another question. What makes the repressor protein? That thing that's upstream called the regulatory gene. Yes. All right, so when the repressor binds to the operator, which is that red arrow, transcription is blank and genes are blank expressed. Okay, so transcription is blocked. And we're going to see how that happens in a minute. And if transcription is blocked, if this repressor protein comes right up here and blocks the RNA polymerase from coming, it's not going to let it happen, right? So these multiple genes are not going to be expressed. They're not expressed because the polymerase can't get to them to read them. So there's a much prettier picture below than mine. So remember, this is the RNA poly. And it's not going to let it go, okay? It's blocked it. It's like a roadblock. So that's how they turn it off. So there are two different kind of operons. There's operons that are going to stay on all the time until you need to turn them off. And then there's going to be operons that are going to stay off all the time until you need to turn them on, okay? So the repressor protein that regulates a repressible operon is translated as blank. You think it's going to be active or inactive? The repressor protein that regulates a repressible operon. That means it can be turned off. Let's write that. Can be turned off. Well, if it can be turned off, then it has to be what? It has to be on, right? All the time, if it has the ability to be turned off. So this repressor protein is always coded at first to be inactive. Inactive. Because we don't need it turned off yet, right? It's just they can be if we need it to. So it says this results in the blank expression of the operon until it is repressed. It says continuous, so it's going to be on all the time. Continuous expression of the operon until it's repressed. So it's going to be, the RNA polymerase is going to be trucking along, reading these genes, making the mRNA, and the mRNA is going to be coding for those proteins. and they work together to produce a product, right? So in simpler terms, the operon is on all the time until it's turned off, right? When the repressor protein attaches to what? The repressor protein always attaches to what part of the operon? The operator. All right, so right now, right, remember, so up here, it's active, the RNA polymerase is reading, we're making proteins, just trucking along, we're in the on position. But hey, maybe I don't need this product up here to be made anymore. Maybe I have too much. So if I have too much, for this example of tryptophan, then I don't need any more, okay? It's almost like a factory. If you think about a factory, my husband works at Lodge, and he makes the Lodge skillets. So they're going to make them and make them and make them until what? He talks about the 10-inch skillet. That's one of their most popular items. So say they make them and make them and make them, and they have 500 back in storage. I'm just making that up. But pretend they have 500. Do we still need to keep production of that 10-inch skillet? No, right? We have enough, so we need to turn it off. So our little 10-inch skillet, or our tryptophan, is going to come be a co-repressor. We're going to have to fill in the blanks about it, so that we can make this repressor protein active, and it can go block transcription. Okay, so once the amount of tryptophan, which is our little 10-inch skillet, accumulates within the cell, it acts as a co-repressor. Co-repressor, okay? And binds to the what? To that repressor protein, but remember, it's usually inactive, okay? It binds to that inactive repressor protein, making it active, okay? So when tryptophan binds, it leads to the conformational change. It changes in shape. Conformational change is a change in shape for these guys of the repressor protein. So you can see that in our diagram over here. It changes its shape to a little square for this representation, which then blank it, okay? It activates it. So that change in shape made it active. So now it can bind to the operator, okay? And it can turn off the operon. This will stop or slow down the production of tryptophan. Remember, that's our little 10-inch skillet, right? We don't need any more. So the skillet's going to go say, hey guys, I don't need any more. Make this repressor protein active so we can block production. That's a repressible operon. Remember, it can be turned off. Okay, that's what repressible means. Has the ability to be turned off, or the ability to be repressed. So guess what? There's going to be an inducible operon. So we're going to write can be turned on. Can be turned on, okay? Inducible operons. Says in blank operons, the regulatory gene codes for a blank repressor protein, and therefore the operon is continually blank or repressed. So in inducible operons, the regulatory gene codes for a active, okay, it's active repressor. The protein, and therefore the operon is continually off, right? It's going to bind to the operator right here. It's in the off state. It's always repressed. It's always off. We're only going to turn it on if we need to. It's like the lights in the grocery stores now. They're trying to save energy, which I appreciate, but all the lights are off until what? Until you walk by and they turn on. It's kind of weird. But they only need to be on if somebody's there looking at it, right? So this active repressor protein is going to block transcription in an inducible. It's always going to be in the off, okay? So it says, so what if we need to turn them on? How do we turn these lights on or turn this operon on if we need to? So guess what? We're going to have a change in shape for that repressor protein. So it says blank molecules attached to the active repressor protein, okay? So it tells us what they are right there. They're called inducer molecules attached to the active protein, the repressor protein, which leads to a conformational change in, I already told you this one, shape, okay? Causing the repressor to become inactive. We're going to pull it off. Inactive, okay? This causes the repressor protein to no longer find, or bind, that's supposed to be bind, I'm sorry, with the operator. And the operator is expressed or on, okay? We're going to pull this repressor protein off. So look at this picture. Here's the example. We're going to pull it off, make it be a change in shape, right? So now the RNA polymerase can go and make its proteins. An example of this is called the lac operon, where lactose in the environment acts as the blank to turn on the lac operon. So guess what? What turned this on? The inducer. Okay, so this is going to act as the inducer. Just like the person walking down the aisle in your local grocery store is acting as the inducer to turn those lights on, right? Same thing happens here.